So if you already have questions that you would like to be discussed actually between also the different panelists, please don't hesitate to put them in the question and answer sections. These can be general questions. This can also be specific questions that you still wanted to ask to specific speakers and that we didn't have the time to answer. So maybe we give another minute or so that everyone is back, but you're welcome already to post your questions, comments, remarks, points that you would like to discuss in the question and answer section. Perfect. It's actually 30 in each of the time. So basically, I was just thinking maybe 4 questions that came up over and over again in the questions and are of special importance. I think one of the most important questions that I think would be interesting to be discussed is obviously we talked a lot about the richness of the microbiota also in African countries, in many countries that maybe have a bit less heavy research on the microbiome for the moment. So one of the questions was, how do you actually make sure that everyone is involved in the research? How do you make actually sure that the genetic richness in these countries is conserved? So I would maybe invite Matthew and also Gloria and Alex to discuss this point a bit more in detail, what is done on the microbiota vault as an example, on the human microbiome conservancy, and what should be actually an ideal way forward to form a global network.
Well, thanks, Pascal. I guess I could start just by saying that the microbiota vault, and I will explain the history tomorrow, but it's an effort that while it will foster research, we will not be doing the research ourselves. It's an effort that points more into preservation, conservation. We will characterize the metagenome of the samples and then open it for the researchers of the world. Also, it's an effort of education, like the GloMiNe that you are attending today and tomorrow. And so rather than a very active research collection that could be global, like the wonderful resource that we saw this morning with Matthew Groussan, it's an active research collection. So there is a difference that make them very complementary. Also with any local collection anywhere in the world, especially in hotspots of biodiversity, which tend to be in developing countries, both environmental and human.
So Matthew, I don't know if you want to comment also for the Human Microbiome Conservancy.
Yeah, I embrace what has been just said and by Maria, and I can also comment on an aspect of your question regarding how we're making sure that the diversity of a country is being preserved. So I will assume here we're talking about the microbial diversity, of course, and so. Two things. So far, what we've done, we collected samples in 2 to 4 different communities in every country where we worked, and we tried to enroll and engage with communities that have very different lifestyles, both urban and rural, but also different ethnic groups, or live in different geographic areas, and all have different diets and lifestyles. So obviously in every country, 3 to 4 different populations is usually not comprehensive. However, what we see based on the sequencing data we collect is that it's a sampling scheme that allows to capture a fair amount of diversity, and we start to saturate in the amount of existing diversity we see by having such a sampling scheme. So obviously it's not comprehensive, but it allows us to capture already a fair amount of the bacterial diversity in our samples. And then it's our work and job to do the culturing work so that we capture this diversity, which we've done fairly well so far, and that we actually preserve it in conditions that allow its distribution for the researcher for like active and live preservation through revival and research.
In that regard, Matthew, can I ask you, there is a real concern, and I'm sure for you too, and it's something that Karina has posted. So it's about how to warrant the long-term preservation of life in the samples and in the isolates.
That is correct. So that's why we're exploring for key species different strategies for long-term preservation. So we're working with we're preserving those bacteria in different states, but obviously there's no state that ensures that the bacteria remain alive for like hundreds of years, and it's the same for those that we actually preserve in stool. So that's why we're actually really trying to push for a model that ensure the long-term preservation of this diversity through active dissemination and regular reviving for sharing with researchers. So we see like previous collection efforts like the DSMZ in Europe as a great way to make sure that this diversity remains alive, right, and preserved. And so dissemination is key here to make sure that it's not dying in freezers.
And maybe taking a question from the chat and expanding it with my own thoughts. So basically, like the question from Bursu Tepekule is how I would like to ask whether there is any consensus on what is and what is not treatable after infancy in terms of microbiome and fecal transplants. And in addition to tie it to what we just discussed, are there also efforts to basically like conserve microbes along the life stage, or are these the same microbes that we find in children and infants and basically in adulthood, or would we actually need to have a collection along the life course?
Pascal, are you asking me? Sorry.
Everyone. So whoever wants to answer can actually answer. Maybe for the collection along the life course, it's mostly you, I guess.
Okay, so very quickly, so far we only recruited adults, but over the next few years, as part of the diversification of the mission we're going to build, it is planned to include infant cohorts and baby cohorts from around the world, and so to start the culturing work from those as well. We know that there are also other groups and researchers who are involved in this and already doing it with some baby communities. And I'm thinking of the great work of Trevor Loyley on this front, who's engaged into this already. So I know that there are ongoing initiatives in addition to what we plan to do that will allow us to get access to this infant diversity.
Thank you.
If I may add, for the microbiota vault, of course, we are interested all ages, not only the gut, but the mouth, the vagina, the skin, not only humans, but other microbiomes associated with animals, with soil, plants, marine, and foods. So far, we are starting the pilot phase with the gut and food, fermented foods, but we hope to grow and really become a backup reservoir for the microbiomes of the planet that are relevant to humans, which are, everywhere.
And maybe just before I give the word to Alex, also like this, I don't know if Martin Blazer is still with us. It's probably a question mostly for him. So he's not. Or then to Gloria, like how much of the microbiome can really be like corrected after infancy?
Question.
Yeah, can you repeat the question? He was away.
Hello. So the question was, what is actually like and what is not treatable after infancy in terms of microbiome and fecal transplants? Was the question from Buiku Tepekule that I probably pronounced wrongly. Sorry for that.
Yeah, so In the experiment that I showed, we treated the mice from day 5 to 10 of life, so very early in life, and then we gave them microbiome back on days 11 through days 13 through 17. So we treated them relatively early. We have not examined late treatment because for this model of type 1 diabetes, the early life events are really critical. By 6 weeks of age, day 42, the phenotype is pretty much set. So we have to intervene very early. Although we haven't done the experiment, from other work, it would suggest that if we transplanted after day 42, it would be too late. So I think in general, this whole issue of restoration after early life antibiotic exposures, timing is very important. And there will be a window for humans. We just don't know what that window is. I hope that answers the question.
Perfect, thanks a lot. Then Alex, I think, wanted to say something. I just got the word that everyone can raise hands. Sorry for that.
Right, thank you very much, Pascal, and thanks to Matthew and Gloria for the initial uptake on that. I'm thinking that this meeting is very important in that it brings us to a point where we need to understand about the agency of preserving the microbiota, particularly with respect to what happens in Africa. Now, it is important to understand that because of the huge diversity, and of course, we all know that the research in the region has not been that much intense when it comes to microbiome research and microbiome activity. Moving forward, of course, with this consortium as a key stakeholder, I'm thinking that if it's possible, we can streamline in terms of methods of collection. I think Matthew touched on that. Well, in as much as that the various type of samples may require different methods of collection and preservation, I think what is really key and important that we have to take note whether we can establish a very simplified protocol that for us, maybe with very small labs, we can all use so that we can have some sort of uniform method in the preservation, so that at least at the end of the day, we might not be wasting our time because somebody collected a sample and treated in a particular way where it doesn't make sense at the end of the day. So because most of the experts are here, I think these are some of the things that I think we should be touching on. And for us, particularly in our region, one of the things that we should be interested in that this platform create an avenue that should help us to connect of course, South-to-South collaboration and begin to connect also very much with the North, so that this kind of easy access or sharing of data or information as we move on in terms of the collection. Because now collection is very interesting, but the preservation, how do you preserve? You may need liquid nitrogen, -80 facility, and some of these things may not be existent. So we collect a lot of samples, but eventually we tend to discard all of them because there's no avenue. How do you preserve sample for how long can you preserve? So these are some of the things that I'm sure that maybe in tomorrow's discussion we may be touching on, but I'm sure Matthew and Gloria has this in mind. And these are some of the things that I think for us in that region, it's very important once we're able to streamline some things. This will be my first initial take on that, Pascal.
Thanks a lot. And maybe just a personal addition, not as a moderator, but as a researcher, also in the Microbiota Vault launch that is currently ongoing, we actually also put efforts on trying to compare different methods to get exactly what you said, Alex, a simple method that will help to basically also revive microbes, hopefully thereafter, to the best extent. Then there was also like Khalid Ibrahim that wanted to add something to the discussion.
Yeah. Hello.
Hello, yes, we can hear you.
Okay. Yeah, okay. I want to thank very much to Matthew for sending me details. So I'm joining to his team now. So we are nearly there to do everything. Just we now focusing to location, which area we have to be collect sample. So this one issue. So as my colleague Alexander, they mentioned how to preserve sample. As you send me email, so you tell me we need to meeting. So I'm just waiting my university to open next week. So I will try to contact. Again, you, Matthew, thank you very much for supporting from Kurdistan, Iraq to support this project for GMBC project. Thank you very much, Matthew.
Thank you. And maybe to change back to viruses again. So there's also a question from Lee Kirchhoff that says, thanks for the excellent talk. We now have the ability to more accurately predict protein structure from the DNA sequence alone. Will having a guess at any 3D structure help resolve what the unknown variant genes are doing?
So unfortunately, no. So you would hope that it would be, but we've just, in fact, I just got one from Rob Edwards, who you guys might know is like the bioinformatics person that I work with most. And he has done a whole bunch of things using that Google's folding thing. And it really didn't do anything for us. The folds that we knew before, we got just fine, but the new folds, especially anything that's associated with the disordered stuff, we got nothing new. So we put about 150 unknown ones through that one. So right now, there's no evidence that the new folding stuff will help get to the virus unknowns. And it is a big challenge, honestly, that we need like a high throughput genetics approach or something in that sense, because we're just left with these unknowns. And it's kind of shocking because it's not just that when we crystallize unknown ones, for example, they clearly fall into real proteins. So it's not that they're just sequence out there in space that's not doing anything. So it's a big challenge. And unfortunately, I don't think that the new techniques are going to fix it. Yeah.
Forrest, I have a question for Forrest. May I ask Pascal?
Yes.
So have you studied what is the effect of urbanization as the diversity of gut bacteria, for example, decreases? Is that affecting the viral diversity and on the gut, so macrophage diversity, but also what is the effect on the host viral diversity when people become depleted in their bacterial diversity?
Yeah, so that's a great question. And roughly speaking, as you kind of, as you lower bacterial diversity, like by a microbialization event, by overfeeding the bacteria, what you see is that you move towards this, the copiotrophic bacteria tend to have a lot of proviruses. So we'll see a switch. Like, I don't know within humans so well because I don't spend a ton of time looking at them, but like on the coral reefs, we'll go from where we have maybe about one provirus per cell out there to maybe 5 or 6 proviruses per cell. And at the same time, we're getting a decrease in diversity of the types of bacteria. So the suggestion is that as we lose the diversity of the bacteria shells, we're gaining diversity in the viruses. That would be my gut feeling / what we've observed in the systems we've looked. I'd like to hear if anybody else has been counting proviruses or anything in different studies. Anybody have any ideas?
Any other virologists out there? Else, I actually have another very difficult question that I would like to ask the panelists.
Still about the virus, tomorrow we'll have a group from South Africa talking about the viral microbiome.
Yeah.
And Ignacio also works in virus. I don't know, Ignacio, if you want to intervene, or we'll wait to hear from you tomorrow. But yeah, I think maybe
Perfect. Then let's go over
Ignacio
to...
is turning on the mic.
No, hi, Karin. So I think I will comment some of this something tomorrow in my presentation. And also Martin Nyanga, that she has been working more on viromes, so on. He will bring some discussion on this as well.
So stay tuned for tomorrow.
Perfect. And this brings us actually to another question that I think is a very difficult question. What is actually a healthy microbiome? Is there actually one healthy microbiome? Are there healthy microbiomes? So I don't know who wants to comment from the panelists on this subject. Karina, is there a healthy mouse microbiome? Maybe this is easier to say than putting in more diversity in it.
Yeah, so I think that this is, I think from a medical point of view, this would be essential to define better. I think this is something we really collectively need to work on. At the moment, we keep using criteria such as diversity, high diversity to define a healthy microbiome. But I think this understanding better what the different classes of microbes are doing will be essential to be able to define what's a healthy microbiome. I think just the definition of high diversity is still very poor, and I would really would like that the field would move forward for a better criteria. But yes, I'm curious to hear other opinions.
I would add to Karina's comment that probably because we are all humans, there are healthy functions, but healthy functions can be performed by different configurations of microbiomes. And the value of diversity is that it provides that redundancy, functional redundancy. So there may be different shapes of communities that provide the healthy functions.
And maybe if I may also add something myself, I think there are probably healthy microbiomes which are adapted. So as we heard, there's a holobiome theory. So I think also microbes are basically part of us, part with us. So probably like the healthy microbiome of someone living in New York is maybe not the same as someone living in an African village. So there's sure like also differences that reflect the greater environment we are in. Any other thoughts on the healthy microbiome? I think the panelists also have to help me a bit because I'm struggling to basically like look what the participants are saying in any of the channels and trying to also like come up with the questions at the same time. So maybe like if you see that someone is asking a question, please just let me know. I think there's also a question from Manuel.
Yeah, thanks, Pascal. Maybe it's a bit of a follow-up to the discussion about the healthy microbiome, but I guess what we learned today is, right, that everyone essentially at the end of the day probably wants to modulate the microbiome in order to achieve a healthy state or a reverse a diseased state. At the same time, we learned that function actually is very personalized, may depend on the virome that you have, may depend on the diet. So I'm just wondering, asking the other panelists essentially, how do you foresee standardized fecal microbiome transplantation, for example, even if it's defined communities, because it seems to me that once you transplant them, they may actually change function again because of the local virome and the diet. So how can we actually achieve rational microbiome modulation that way?
So I'll jump in that, really when we as a group, and we're thinking about this, we're going to have to embrace a very much more ecological viewpoint of it, right? So you're kind of steering a boat where you're going like this. It's not like we're going to just come up with something that works and is a magic bullet for everybody. And in fact, it's probably that it won't be a magic bullet for even 2 people. We're going to have to probably do really truly personalized things and say, this is what we're trying to drive this direction with this particular person, and what is predictive to allow us to do that. Does that make sense? Like, because we can do that in some diseases now. And I think we can, if we view health as some state that's not dissimilar from a disease, right? We're pushing it one direction, then that may be the best way to get there.
Yeah, I think that makes sense, Forrest. Maybe just as a follow-up, but what you're saying then is that essentially you would need to do kind of a multi-omic screening of each individual person before you can come up with a strategy that works. And how could that ever become economically feasible?
Yeah, so I think that's a great point. So we, you know, so I'm just going to go back to our stuff just because that's what I know. So with cystic fibrosis, like we can do that very nicely now, where we kind of say, this is what's going on with the person. And you're correct, you need like at least 2 samples to get you any idea where you're headed. And then you start manipulating it, you know, you do things to the patient and you see how the system responds. And that used to, of course, be, you know, a $20,000 job. Now it's probably a $200 job. So we're getting better at it. It's still mostly bioinformatics that is slowing everything down and having the models to compare against. So I think eventually you'll have it where it'll be cheap enough. Maybe we don't have to do all the fancy things, right? Maybe we don't have to sequence each time. Maybe the metabolomes will be good enough once we understand the metabolomes better.
So maybe I'll just give the option of I'm sorry, I don't know the name, Briho from the audience to speak. Yes, you can talk.
Hello.
Yes, go ahead.
Yeah, good afternoon. Hello. Yeah, I think I just wanted to know the impact on the especially for example E. coli on a specific virus, on the vice versa. For instance, what would be the immune response from these microbiomes, E. coli on a specific virus in the gut or the GIT, and vice versa with all other microbiomes being not coming into play, for instance.
So the question is, when a new bacteria comes in, what is the impact on the viriome? Is that right?
Yes, yes.
Mm-hmm.
Yes.
Yeah, so that's probably the way that most of the viruses actually make it into the gut after, or at least in humans, after differentiation of the gut. It's very hard for a virus to make it all the way down to the colon. So probably most of them are being delivered via bacteria that are good at invading into the gut or into the colon. And so they bring phage, we know that. And then we know sometimes the phage can kill other things in the other bacteria that are related in the gut. So you can see small changes in the microbiome based on that. You would imagine that that there are pretty dramatic changes. So one thing to remember, it's a little off topic, but most, if not all of the bacterial pathogens, let's say 90% of the bacterial pathogens are actually bacterial pathogens because they're carrying the proviruses, which have the functions that lead to the pathogen doing its thing. And in the cases where that's not true, that most of the important ones are usually on something similar like a transposon or a plasmid. So there's pretty dramatic effects of having this happen, but I can't answer anything specifically with E. coli or something of that nature. And I'm talked too much, so someone else needs to talk.
Thanks a lot. So maybe we can just take a last discussion point. We're almost running over time already. So Karina Dur is actually asking a very important question. What is actually the influence of climate change on the microbes profile? So I think that's a very difficult but very important question. So I don't know who wants to comment on it. I think there will be several things happening, obviously. So Matthew, I see your camera is turned on.
Yeah, I think one comment we could make is so there's been little association between latitude and microbiome compositions or diversity. It's not like a factor that is driving much differences in microbiome between people. So elevated temperatures might not have a big impact. However, what will likely to have a big impact is changes of lifestyle due to populations that have to move from rural areas to more urban ones, or changing of geographic areas due to migration. And this will very much likely have the biggest impact on the microbiome. And at least that's my prediction. So it's more like an impact on the microbiome due to the consequences of climate change, and that might not be due to just the rise of temperature itself.
Perfect. And I think this is actually, if there's no one that has a burning point to raise, I think that would be maybe a nice point to also close the discussion. From industrialization to climate change to the fact that actually we really need to preserve the microbiome, that we need to preserve it on a global scale. So I don't know if Karina maybe wanted to say something also for the session of tomorrow.
I was confused if you were asking Karina or Karina. But yeah, so yes, because, okay, so yeah, for tomorrow, just come back, please, for tomorrow. Remember that we'll start 1 hour earlier, which is a bit difficult for some people, a bit easier for others. I think this was a great discussion. That there were still some points about antibiotics that I was being asked during the chat that we can come back to tomorrow. There will be discussions about virus again, and lots of good examples on African research stories, and new technologies on how to analyze the microbiota. So looking forward for tomorrow. And thank you very much. I think it was a very active session. We've tried to manage answering all the questions. It's not always possible, so but keep participating. Thank you, all the speakers, and to those that had to wake up very early. I'm sorry about that, and I hope you can rest now. Bye-bye.